Have you ever thought about what's happening inside you right now at a microscopic level? Let's take a journey. We're going to zoom in past your tissues into a single one of your trillions of cells and go right to the heart of the action, the nucleus, because that's where our story begins, with the incredible, intricate dance of cell division.
So here's our game plan. First, we'll look at the cell's life calendar to get the rhythm of its existence. Then we'll dive into mitosis, which I like to call the cellular photocopier. After that, we'll explore meiosis, a totally different process that's all about shuffling the genetic deck. Then we'll compare them, and finally, we'll get into how this whole system is regulated and what happens when things go wrong.
All right, let's kick things off with Chapter 1, the cell's life calendar. Before we can understand how a cell divides, we got to understand how it lives. What does a day in the life of a cell actually look like?
But let's start with the most basic question of all. Why bother? I mean, this is a really complex process. So what's the big deal? Why is cell division so absolutely fundamental to life as we know it? Well, it really boils down to three huge jobs. For a single-celled organism like bacteria, it's pretty simple. It's how they reproduce. For complex critters like us, it's how we grow from a single fertilized egg into, well, you. And it's also how we stick around, constantly repairing our bodies by making new skin cells, new blood cells, and so on.
Check this out. This visual gives us a great look at that life calendar. And the first thing that probably jumps out at you is that a cell spends about 90% of its time in what's called interphase. Now, don't let the name fool you. This is not a resting phase. It's go time. The cell is growing, replicating every single strand of its DNA, and getting all its gear ready for the main event, division.
Okay, before we go any further, let's do a quick vocab check to make sure we're all on the same page. A chromosome, that's just DNA that's been tightly packed up for easy transport. Now, most of your body cells are diploid. Di means two. They have two sets of chromosomes. In humans, that's 46. But your reproductive cells, your gametes, are haploid. They carry just one set, or 23 chromosomes. All right, let's jump into our first type of division, mitosis. And the best way to think about it is as a perfect, high-fidelity cellular photocopier. So, what's the mission for mitosis? It's simple. Duplication. The whole point is to produce two daughter cells that are perfect genetic clones of the parent cell. This is absolutely critical for growing and for repairing tissues. You want your new skin cell to be a skin cell, right? That's what mitosis ensures. Now, this copying process isn't just chaos. Oh no, it's a beautifully choreographed dance with 5 distinct stages. We've got prophase, prometaphase, metaphase, anaphase, and telophase. Each one makes sure the DNA gets sorted out and delivered perfectly. Let's break them down. First up, prophase. Imagine all that loose, stringy DNA, what we call chromatin, suddenly getting super organized. It coils up incredibly tightly into those classic X-shaped chromosomes that you can actually see under a microscope. At the same time, the cell starts building the machinery, the mitotic spindle, that's going to pull these chromosomes apart. Right after that, in a quick but crucial step called prometaphase, the nuclear envelope dissolves, allowing the spindle fibers to grab hold of the chromosomes. And that sets the stage for this beautiful moment, metaphase. Just think M for middle. Look at that. Every single chromosome lines up neatly right down the cell's equator. It's a perfect formation, ensuring the split will be completely fair. And then, bam, anaphase. Think A for apart. The sister chromatids, the two identical copies of each chromosome, are ripped away from each other. The spindle fibers yank them to opposite ends of the cell. It's this incredibly decisive moment where each side gets a full, complete set of the genetic blueprints. And in the final act of this nuclear drama, we have telophase. The chromosomes arrive at their destinations, the cell starts building brand new nuclear membranes around the two sets of DNA, and the chromosomes themselves start to relax and unwind. But hang on, we've got 2 nuclei in one cell body. That's not going to work. The last step is cytokinesis, the actual splitting of the cell. And it's not a one-size-fits-all process. Animal cells, like ours, basically pinch in the middle, like pulling a drawstring. But plant cells have a rigid wall, so they can't do that. Instead, they cleverly build a new wall called a cell plate right down the middle to divide the space. Okay, let's totally switch gears now. We've seen the photocopier, mitosis. Now it's time to meet meiosis, which isn't about making copies at all. It's about shuffling the genetic deck. So what's the point of this whole other process? Well, meiosis has a very specific and very important job: to make the sperm and egg cells needed for sexual reproduction. And the key word here is uniqueness. It starts with one diploid cell and ends with 4 genetically different haploid cells. The real magic, the source of all this amazing genetic variation, happens right at the beginning, in Prophase 1. Here, the homologous chromosomes, one you got from your mom and one from your dad, find each other and pair up. Then, they do something incredible, called crossing over. They literally swap pieces of their DNA. It's like they're trading genetic secrets, creating brand new combinations of genes that have never existed before. How cool is that? So meiosis is a 2-act play. Meiosis 1 is the big one. It separates the homologous pairs, which cuts the chromosome number in half. Plus, there's another shuffle here called independent assortment, which just adds more randomness. Then meiosis 2 is basically a quick round of mitosis. It just separates the sister chromatids. The grand finale? 4 totally unique haploid gametes, ready for action. Okay, we've covered a lot of ground. Let's do a quick head-to-head comparison now, just to make sure we've got the key differences between these two processes locked in. This table lays it all out perfectly, doesn't it? Mitosis, 1 division, 2 identical diploid cells, 0 variation. Its purpose, growth and repair. Meiosis, 2 divisions, 4 unique haploid cells, and a ton of variation. Its purpose, sexual reproduction. They are 2 completely different toolkits for 2 completely different biological jobs. Now, with all these moving parts and all this complexity, you have to wonder, how does the cell keep this all from going haywire? And, what happens when it does? Well, it turns out the cell has an amazing quality control system. You can think of it like a series of traffic lights or checkpoints. At each one, the cell basically asks itself, hey, is everything good to go? Is the DNA copied correctly? Are the chromosomes all attached? And if the answer is no, it hits the brakes. The whole process stops until the problem is fixed. And this is where it gets incredibly relevant to our own health. You see, normal, healthy cells respect these checkpoints. They stop at the red lights. But cancer cells? They've essentially cut the brakes. They blow right through all the stop signals and just divide, and divide, and divide. That uncontrolled division is the definition of cancer. Errors can also pop up in meiosis, and they can have really big consequences. Sometimes chromosomes fail to separate properly, a mistake we call non-disjunction. This leads to gametes with the wrong number of chromosomes. And as you can see in this karyotype, if that gamete is part of fertilization, it can result in genetic conditions like trisomy 21, which is an extra copy of chromosome 21 that causes Down syndrome. And that really leaves us with something to think about, doesn't it? The cell cycle is the engine of life, of growth, of everything that we are. It's just amazing to realize that the health and future of an entire, complex organism hinges on the flawless execution of these tiny, microscopic, molecular steps. It's pretty incredible.